Hello and welcome to another episode of our oncology podcast series, Onc Now, with me, your host, Dr. Jonathan Sackier. This series spotlights the leading experts in all subspecialties of oncology, and today I'm really delighted to be joined by our distinguished guest, Professor Sarah Blagden. Sarah completed her bachelor's in pharmacology at the University of London and her medical degrees at Charing Cross and Westminster Medical School, and then later a PhD in genetics at the University of Cambridge. And she also pursued a clinical fellowship at the Institute of Cancer Research Drug Development Unit. Currently, Sarah holds the position of Professor of Experimental Oncology at the University of Oxford, where she leads the Experimental Oncology Research Group. As if this wasn't all impressive enough, Sarah has also found the time to be CEO and founder of RNA Guardian Limited, a biotechnology company dedicated to identifying early cancer biomarkers. Beyond her professional commitments, Sarah likes to spend time with her family, especially her 2 children, and is an avid reader. So, without further ado, let's head over to the podcast conversation with Professor Sarah Blagden. I hope you enjoy. What have you read recently that's tickled your fancy, and might we expect to see your name on the spine of it? Oh, absolutely.
So what tickles my fancy? And thank you for having me on the show. What tickles my fancy? I love a mixture. I think I quite like a mixture of kind of fiction and non-fiction at the same time. So I quite like
I think you read like multiple books, have something going. Yeah, I've got like 9 on my bed.
Yeah, I know, and there are certain moods to certain things. So, for example, I'm reading a lot of Maggie O'Farrell. So there's a book that she's written called The Marriage Portrait, and it's all about Renaissance Italy, and it feels very claustrophobic and threatening. And then at the same time, in parallel,
that's historical fiction.
It's historical fiction, but it's it's really all about, all the different warring Italian houses, which led to the Medicis and all that sort of stuff. So that's kind of interesting because it's an area I don't really know much about. And I find myself then going down these sort of Google rabbit holes, reading around it. And then at the same time, I quite like a bit of kind of science, popular science. I've read a lot of books like, for example, Nick Lane. I don't know if you've read his works, but it's really interesting. There's one book was incredible called Oxygen, and he really describes our relationship as an organism with oxygen as we've evolved, and how fundamentally our bodies are trying to resist rust. But we'd like an apple core going kind of, that is what a lot of our cellular functions are all about. It's kind of surviving in an oxygenated world. So that kind of thing I really enjoy. And I've been reading a lot of things about aging as well, because I think there's really nice parallels with cancer and aging.
Right. So I'm with you with that kind of book. I read one recently, The Disappearing Spoon, and it's an explanation of the periodic table, element by element, and disappearing spoon. I think it's it gadolinium. It's one of the metals. Apparently, physicists, materials scientists like to make spoons out of this stuff and giggle crazily when they give you a cup of tea or coffee and you put your spoon in it. It melts. It's got a melting point of, 40.
How amazing.
But it really, I think it opens your mind, as when we work in a particular area of science, we don't think about other areas of science. But
I agree. And didn't the periodic table come to him in a dream? I think he had a dream and then woke up the next morning and wrote it all out.
It started.
Boring, I thought.
Yeah. Putting it into the different
columns. I saw them all come into today, and it's just...
It's a fascinating read. Highly recommend.
Yeah,
so
it's really good.
Like we see your name on the spine of the book.
I feel really caught between a love of fiction and this. I love fiction because you kind of live someone else's life, for a bit. Particularly if you go to sort of full immerse or holiday or something like that. But then I also like little facts I don't know. I can't find the genre that would suit me.
Yeah.
So
medical textbooks are, I don't know, about a labour of love, a labour of hate. I've done a few of them, and
I've done a few chapters like that.
Yeah,
talk show.
It's absolutely,
because every single word has to be checked to the deal.
Exactly. Yeah. It's not the freedom of writing fiction. You also mentioned that you tweet. What do you tweet about?
I tend not to tweet particularly politically, but I tweet things that have struck me. So, for example, I tweeted last year about TB, because it struck me that one of the things that, as a cancer doctor, I find is there is a kind of cultural acceptance of cancer, which I find odd. I feel like we should be doing more, not just to treat established disease, but trying to understand the origins of cancer, how it starts in our bodies. But I think because when people say to me, well, cancer is just really something that has co-evolved with human beings. It was around since the Egyptians, they found cancer deposits in bones washed up on the lake or whatever. And I find that frustrating. And a similar attitude was abundant around the time of TB during the worst of its kind of European epidemic in the 1900s, early 1900s. There was a similar perception. And it was just felt to be the disease, a price you paid for a lifestyle.
Slightly inevitability.
Inevitability, exactly. And interestingly, when Robert Koch actually identified it was a bacillus that caused it, that it was a bacteria, there was a huge amount of education to tell everyone to go and start antibiotics, to take part in TB screening, because there wasn't an incentive to do that. There wasn't the belief. And there are all these posters really warning people that, that there's one path where you can live and another you can die, but you need to go down the live path.
Yeah. This is like a cultural imperative. I mean, during our lifetimes, the way that breast cancer, I remember in polite society, you didn't discuss it. You sure as hell didn't have articles about it in women's magazines raising the profile. Recent television advertisements advising men to do testicular self-exam, and then you gradually erode these belief systems. Of course, historically, leprosy, it's unclean, you offended God.
Yeah.
So, there's cultural changes.
And there still are cultures where they still have that stigma around cancer.
Yeah.
And if you go into, say, an ultra-Orthodox Jewish community, there is a kind of sensitivity about whether or not you're a BRCA carrier, for example, which is a gene mutation that does predispose you to cancer. Yes. And so there's difficulty in discussing that. And again, that really thwarts efforts to really address cancer if we have those kind of pockets.
Right. So tell us about your journey into oncology. What took you into research and your motivation to transition from clinical practice to focus on translational research?
I think I didn't think of myself as being particularly academic at medical school, but I was very struck really by oncologists. The oncologists I met and worked with, there was a guy called Bob Phillips, who was wonderful. We used to do ward round. I sort of followed his team around in the old Westminster Hospital on Horseferry Road. And he was just so incredible with his patients. He was just so lovely. And they had so much confidence and faith in him. But at the same time, he was brilliant at the science. He understood it all. And I just felt that was one of the few specialties that really asks two extremes really out of you. It needs you to be a kind of almost like a psychologist and be someone who's comforting and be someone who's, knows the answers, or at least, knows or at least seeks the answers. And yet with a very strong foot in the science. And so I decided that was kind of something I really wanted to aspire to be. And at the same time, my own grandmother, she was in her early 70s, had cancer diagnosed. And at that time, it was colorectal cancer. It was another one you didn't mention. And they said, well, there's no treatment for you. They didn't advise any treatment. And she was sent home to die. And it was it was horrible. And it just felt, even then, it felt quite paternalistic. And it felt like we must do better than this. And then as I sort of got interested in running trials or not running them, but, helping people run trials when I was a junior doctor. And I kind of didn't really like the trials. I thought we can do better. This should be more interesting, And so that's kind of how I ended up going towards research, was I wanted to sort of ask the questions myself.
What you find more interesting?
Well, there's a lot of work that we do around clinical trials, which is maybe asking a question that's posed by pharma. It might have a new drug that's slightly different to the last one, but, it's having a very incremental effect on cancer outcomes. And I really wanted to explore new drugs or new ideas or understand the biology in sort of from a different perspective. And so that's how I ended up thinking, okay, well, maybe I do want to do a bit of research. And then when I was in Cambridge, I had this wonderful mentor, Helena Earle, And she sent me off around the labs to go and talk to, different teams and just find out what interested me. And I really got fascinated by a fruit fly lab. And that's kind of how I ended up doing research in Drosophila. And really that kind of led to me being interested in RNA biology and where I'm really now.
It's a very, very common theme that I hear when I talk to people from different specialties. There's always one or two people that you meet at a pivotal moment in your life. For me, it was a surgeon who just had that persona, that warmth about them, and the way they dealt with people with serious disease, and imbue them with confidence, whilst not coming across as, you use the term, paternalistic, which sometimes we, especially surgeons, get accused of.
I know, seeing that sort of professional kindness is amazing. Someone can go be so, you're very senior at the time and still has retained that touch of kindness. It's lovely to see.
And I certainly always talk to my residents and tell them that, remind them of the words of Osler, that a good physician should be able, which is obvious, available and affable. And, if you're not the latter, well, I don't care how technically good you might be, because you won't win the hearts and minds in the minds, but not the hearts, for sure.
He's incredibly holistic.
Yeah, very much so.
And it's quite unusual for his time, I think.
Yeah, the latter part of the 19th century, early part of the 20th, I guess. So you've worked at various institutions and seen various people. How's that shaped your thinking? I want to come back to clinical trials, because as you're answering the first part of the question, please think about that. I think we need to completely change the way we view clinical trials for many reasons, not the least of which, especially you're dealing with your specialty with people who have a fatal illness, right? And the amount of time it takes from the idea to a new drug or a therapy being licensed is way too long. What can we do to speed it up? So your experiences at institutions, and how can we improve the clinical trial environment?
Yeah, so my experiences of institutions, it's interesting. I'm a clinician scientist, so I kind of wear both hats. And every institution has its own kind of innate preference for you as either a clinician or a scientist. And you have to actually find the institution that kind of suits you rather than vice versa. You might go to one place and they'll say, we really want to give you research time. Then you find that actually you only have 20% time to do that. And you find yourself being pushed and being sort of feeling constantly guilty that you're not doing enough from the clinic. And then in other contexts, it might be too much emphasis on research and not enough respect for the clinicians. So I sort of moved from place to place to find, I as part of my kind of training, I moved around. But I think for me, where I am right now in Oxford is quite nice. There's a really good balance. There's a very healthy balance of at least sort of 50-50. And that's kind of like I find myself veering, I suppose, more and more towards academia now. And yeah, I'm also, I share your feelings about clinical trials where they are too slow, particularly in the area that we're looking at, which is going into sort of cancer prevention trials. They can take, they recruit thousands of patients. They take, on average, between 10 and 15 years to run. There isn't any biology in them because you can't take samples from that many patients. And so you end up with a result, an outcome in the trial where you're kind of left not really understanding any nuance in the data. And sometimes you're no better off than when you started. And I think we have to sort of think about rejigging the way we run, prevention studies and start making them much more biological and much more kind of targeted, much as we've done in sort of oncology these days.
Mm-hmm. So looking at your research career, it began with a Cancer Research UK Junior Clinical Scientist Fellowship. For your PhD focused, as you said, on Drosophila and fruit fly genetics. How has your background in genetics shaped your current work in oncology? And I know there have been enormous advances in breast cancer, for instance, we were talking about another doctor who I spoke to. So yeah, tell us about that.
Well, I think up until then I thought that research was a very other thing, particularly fruit flies, because it seems a little bit esoteric. But funnily enough, one of the things that came out of the lab while I was there, and actually most of the work had been done way beforehand, was work on a kinase called polar kinase. And when I started work in the phase 1 unit at ICR, the Institute of Cancer Research, with Johan Bono and Stan Kay, who were very sort of prominent phase 1 oncologists. And one of the first drugs that I started working on was actually a polar kinase inhibitor. So it felt very personal because this was, I knew the team that worked on that in the lab. And it made me realize that actually the steps between research and basic research and clinic can be quite short. And it sort of really woke me up to this idea that I could get involved more in these early phase trials. And it gave me the feeling that I was kind of as near as I could be to the lab. And then I actually started my own lab. So I had a nice kind of triumvirate because I had the clinical side, I had my own lab, and I had the trials. And that was why I was at Imperial.
And you were doing work on post-transcriptional gene regulation.
Well, it's really about, we know we've been taught that cancer is a disease of the genome, but we know that there's probably a lot more to it than that. So, you can have multiple mutations in completely normal cells. Particularly as you get older, you're more likely to have, quite a lot of mutations in your normal cells. That doesn't necessarily mean that you're about to sort of start a cancer. So clearly something else or multiple other factors are kind of conspiring to turn those mutated cells into cancer. And so one of the aspects that my lab has been working on is RNA binding proteins. So these are proteins that are essentially bound to the messenger RNA, which is the middleman between the gene and the protein, and are actually sort of distorting the message. So whereas the gene might have wanted to encode maybe one copy of mRNA, it doesn't at the end of it synthesize any protein. Or you might find the opposite. You might find one copy of messenger RNA creates tons and tons of protein. And that's because of the distorting effect, really, of these RNA binding proteins.
Fascinating stuff. So at Imperial, you launched the early phase trial portfolio and established a lab focused on dysregulation of mRNA translation in cancer. So I presume that led to this work.
Yeah, so and that was one of the things that we did was really clone the human homologue of the fruit fly gene that I've been working on. So we described it in humans. It was called LAP1, and then we were able to show in cancers that it was highly overexpressed, highly upregulated. And then we tried to understand why and what it was doing. And then that story has kind of led into Oxford, into the research I'm doing in the lab now, has moved us into metabolism, thinking about the cell's metabolism, and starting to think about how that gets distorted during that transition between normal cells as they become cancerous, so tumorigenesis or carcinogenesis. Metabolism goes wonky at that point, and we're trying to understand why and how.
Right.
And is it being driven by these RNA binding proteins?
So I know that the work is focused on precision prevention.
Yeah.
Right? And you've hinted at the amount of time these studies can take. And given the fact that we've got an expanding population, diminished resources, the cost of care is going through the roof. Every country, every first world country is suffering and facing an uncertain future. Prevention seems to make more and more sense, right? And, obviously it can limit the morbidity and mortality. So what seeds of hope are you seeing? What?
Well, one of the yeah, well, I think for me, I was always quite shocked and surprised to discover that cancer, doesn't just strike without warning. It actually takes quite a long time to develop in an organ. So, for example, pancreatic cancer can take up to 15 years to develop. Colorectal cancer, 20 to 30 years.
Yeah, polyp cancer, that's what we've known about for decades.
Exactly, and we're able to see the polyps, so we have at least the chance to intervene. But for many of those internal organ cancers, you can't see a polyp. You can't see them on CT scans. You can't detect them on blood tests at the moment. So for me, the challenge is really trying to expose those, trying to identify ways that we can detect them. But what is really interesting is during that latent phase, during that kind of gestation, those cells, they're not cancerous, they're not normal either. They're in that kind of middle ground. They don't release the same biomarkers. They don't kind of behave in the same way as a cancer. But one thing that's very different is they're actually reversible at that point. So you can reverse them. In fact, our bodies are probably always doing that. They're probably always reversing these pre-malignant lesions. And so in some cases, they get through. They get through our immune surveillance. They're not reversed. Yeah, they have a cloak of invisibility. They're not reversed. And so what we're trying to do now is develop vaccines and things that we can use to actually kind of force the immune system to recognize and reverse these lesions. And so that's kind of led us to thinking about, okay, how do we trial? How do we run studies of these vaccines or of these targeted drugs? And obviously we don't want to run them over hundreds and thousands of people over 15 years because we'll really never get anywhere. And as you say, there is an encroaching epidemic of cancer. So instead, what we're going to be doing is running these really sort of tight, randomized, controlled studies in high-risk populations. So we identify a high-risk group. We've talked previously about BRCA carriers, for example. There are communities, for example, with Li-Fraumeni syndrome, which is you have a 90 to 100% loss. Yeah, the risk of cancer, 90 to 100%. And they've often been excluded from studies because they are their risk is too high. It's not typical. But why don't we turn it on its head and actually focus our research on those high-risk groups who have the most to gain from an intervention that works? Yeah. And then because it's smaller numbers, we can do quite a lot of biology on them. We can take blood tests, we can assess the ctDNAs and biomarkers and so on. So not only are we looking at testing and intervention, but we're also understanding the tumorigenesis process at the same time.
Yeah.
And the time frame can be shorter.
We've seen, haven't we? Like, obviously, in my work in surgery, the polyp cancer sequence, and seeing people who've got familial polyposis and know with certainty they're going to get a colorectal cancer, and we can do something about it. And of course, HPV, prevention of cervical and laryngeal cancers with the vaccination. So there is some promise, and there is a model that this can work. You mentioned, Li-Fraumeni, you published an article reporting on a randomized controlled trial looking at the use of Metformin, which I know that people in the United States buy it and think it's the new vitamin M. Right, everyone's taking that as well as the GLP.
Well, that was the only third. So you see the paper in Cell that was showing in monkey brains. They gave monkeys metformin for 3 years and demonstrated that it had an anti-aging effect.
Yeah.
And quite significant. So I think in the monkeys it was about 18 months, but that translates to 10 plus years in humans.
So we should all be taking metformin.
Well, it has side effects. It is quite a dirty drug. It's a natural. It's derived from French lilac. It's a natural drug. But interestingly, so I talked before about designing studies in high-risk groups and trying to match them to, or trying to identify. First of all, you have to identify a sort of biological vulnerability in that condition, and then you try and match it to a targeted therapy. And in the case of Li-Fraumeni, one of the research that came out of this guy called Paul Huang's group, he's now at the ICR, was to show that they actually have abnormal mitochondrial metabolism, so that they are produced, their oxyphosphorylation rates are very high. So their mitochondria are really churning out a lot of reactive oxygen species, which are kind of these dangerous free radicals that go in and kind of destroy and cause mutations and so on. And they demonstrated that, or rather he demonstrated that metformin, amongst its myriad of mechanisms of action, actually seems to inhibit that oxyphosphorylation, and others have shown that it acts through complex 1 of the electron transport chain. So basically it's right in the mitochondria, kind of turning it down. And so they showed in mice that it prevented those Li-Fraumeni mice from getting, or delayed them from getting cancer. So what we want to do is really test that in a Li-Fraumeni population. And again, what we also want to do is take samples from the patients and really go back to the basic biology and try and understand why cancer happens in that group. We know that they carry mutated P53. But again, back to the previous point, just having a gene mutation isn't enough. What else is going on? And that's kind of what we want to understand. So yes, metformin's fascinating.
the potential for it to be a widely or to be the basis for work to lead to a widely used cancer preventative.
I mean, they've shown in a large, there's a big meta-analysis of, I can't quite remember, I think it was like 3 million people or something who have been on metformin. They were diabetics though, and they showed that they had a lower risk of cancer than people who were on insulin.
Right.
But that is not a kind of normal population, and I don't think any of the data in normal populations using metformin has really shown be quite so impressive. So I think metformin may not be a useful cancer preventative, unless you have a specific vulnerability that it's affecting, if that makes sense. So I don't know, but I think there must be an element of metformin which is the key to its success, and that's it.
Yeah, it actually raises another point. It's like the GLP-1 drugs that there's been a lack of availability because anyone who's got a couple of extra pounds is getting hold of it. Apparently, I was in LA recently, and a colleague there was telling me that he says, look around. He says, you want to see GLP-1 in action? Here it is. You get them up.
But it's interesting because there's also an anti-cancer effect with GLP-1 inhibitors. So we're seeing this convergence of biology, which I find out fascinating, which really goes back to the anti-aging and all of that sort of thing. There's something interesting that in the end, I think we're all going to be working together.
Yeah. Folks, I hope you enjoyed that first part of my conversation with Professor Sarah Blagden. Tune in 2 weeks for the continuation of this conversation. Meanwhile, please subscribe so that you never miss an episode. Check out the archives, like us on social media, you know the drill, and tell your colleagues. Until next time, thanks so much for listening, and please stay safe, stay well, stay curious. Bye for now.